Welcome to the Australian Biocommons webinar series. My name's Christina Hall and I'm the Australian Biocommons Associate Director, Training and Communications, and I'll be your host for today's webinar. I'd like to wish you all a happy International Women's Day and welcome you to reflect on the many great women who inspire us with their contributions to science and bioinformatics. And I'm delighted to say that you'll hear from one of those women today. In this series, we share useful information about the latest digital techniques, data and tools for the life science community. Each month, we hear from local and international experts who present a bioinformatics topic that we hope will support Australians to deliver their best environmental, agricultural and medical research. You can keep up to date with the latest Australian Biocommons news and events via the channels listed on the screen here. Before we begin, we'd like to take a moment to acknowledge the traditional owners and their custodianships of the land on which we meet today. In my case, I'm in Canberra on land of the Ngunnawal people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. We particularly like to welcome those of you joining us live today, and to help you get the most out of the session, here are a few things to be aware of. The session is being recorded and you'll find it soon on our YouTube channel, along with recordings of previous webinars for you to browse through. Auto-generated captions are available during this webinar, and you can turn those on and off using the live transcript function in the Zoom dashboard. You'll have the opportunity to ask questions via the Q&A function on your Zoom dashboard, and these will be addressed towards the end of the presentation. Today, we're thrilled to welcome Dr. Carolyn Hogg to speak to us about conservation in the age of extinction. Carolyn is a senior manager with the Australasian Wildlife Genomics group at the University of Sydney. She's been working on the conservation of threatened species for over 25 years, both in Australia and overseas. And Carolyn's vision is to work closely with academic and conservation management partners to create a conservation legacy for Australia by changing the way we integrate science, management and policy to proactively promote species resilience in the face of a changing world. Today, Carolyn will share insights into how this vision is being put into action to build genomic resources that will help us to understand and protect Australia's threatened species via the Threatened Species Initiative. Welcome to the webinar, Carolyn, and I will now hand over to you for your presentation.
Great, thanks so much, Christina, and thanks Biocommons for inviting me to speak here today. So hopefully that's come up so everyone's able to see it. So first of all, I'd also like to take the moment to acknowledge the traditional custodians on the land on which we all meet today. I'm on the land of Wallumedegal people here in Sydney, and just to acknowledge the elders past, present and emerging. I'd also like to acknowledge International Women's Day. We have a very large team of our team here at the Australasian Wildlife Genomics Group, and predominantly female scientists. So kind of big shout out to all those women who've contributed to our research over the years. So today I wanted to talk about conservation in the age of extinction and give you a bit of insight into how we're using the latest genomic sequencing tools and also bioinformatics tools through the Biocommons to help facilitate some of the research that we're doing here in Australia. So it doesn't take anyone who hasn't seen the news of late. However, it seems to have dropped off the radar, but with the extensive rain we're having here in Sydney at the moment, climate change is having a pretty significant impact on the planet. But in addition to that, the other side of the climate change story is really the story about biodiversity loss. And we're now found ourselves in another period of mass extinction, and we're losing biodiversity at an exceedingly rapid rate. Recently, a couple of years ago, the international UN, which is the IPBES report, which is very similar to the IPCC, which is the climate change report, this is the biodiversity report, declared that at least 1 million species are now at risk of extinction. And that's because we've seen a significant change to land area use, as well as a loss of wetlands, impacts on the ocean environment, as well as the impacts that we'll have projected on the different types of land use that we have at a global scale by 2050. And a lot of people often say to me, well, you know, why is biodiversity so important? Why is it so key? And it's not just about having a nice environment or having lots of species in the environment when you go out in the bush or go swimming in the ocean, But what we do know is biodiversity is really critical to our survival. We know that it is linked directly to human livelihood and human sustainability around the food resources that we have. It has both cultural and economic value and importance around a range of different activities, from energy to recreational activities. It's also very, very important to our health. The global COVID-19 pandemic is a key example of the impact it can have on health when you cause individual species that have not co-evolved together to live very closely together in environments where they traditionally haven't come in contact with one another. And also, it's one of the greatest solutions for climate change, and that's really around water recycling, improved microclimates, and also the large amounts of forestry and land clearing that's going on is exacerbating the heating of the planet. So biodiversity is really important, not just to the planet's survival, but it's really important to our own survival. And if we look at Australian biodiversity loss, the recent OECD Environmental Performance Review that was published in 2019 has actually showed that the status of Australian biodiversity is very, very poor and is actually worsening. And not only that, but the cost of Australia not meeting the Paris Accord between 2020 and 2050 is close to $1 trillion. And that $300 billion of that comes from biodiversity and human health. And so not only is it a social and environmental loss with biodiversity, but there's going to be pretty significant economic consequences to us losing biodiversity over time as well. So what does this actually all mean? So when we talk about biodiversity, biodiversity is really underpinned by three pillars. The first pillar being biodiversity of ecosystems. So the more diversity of different ecosystems that we have across the planet, the better off our biodiversity is. We also know a lot about diversity of species within those systems. The more diversity of species we have, the greater those systems are at functioning. And for a very long period of time, ecologists have been very good at studying both ecosystems and species and understanding their contribution to biodiversity. The third pillar that underpins biodiversity, however, is diversity of genes. And we've known that the genetic diversity is important for a long period of time. We've really tried to incorporate conservation genetics and genomics into the conservation management space, particularly in the last decade. But it has been quite difficult to include diversity of genes into the long-term studies of biodiversity. And for that, that simply comes down to the lack of technology that existed at the time that all the other studies were going. But the age of genomics really is upon us. Although it started back in the 1960s with studying very, very small genomes of E. coli and yeast, it wasn't until the Human Genome Project in the early 2000s that we really saw a massive change in uptake in the genome sequencing technology. And that's, of course, observed quite succinctly when you look at the very large increase of genome sequences that have been inputted into like the national repositories such as NCBI since the Human Genome Project, and also very fast acceleration in the types of medical research in particular that have been able to be undertaken because of the Human Genome Project. In 2018, the Earth Biogenome Project was launched, and the Earth Biogenome Project is a global program to try and help facilitate and generate genomic resources for all eukaryotic life on Earth. And so the Threatened Species Initiative, of which I'm the science lead and going to talk a little bit more about today, is part of that larger umbrella Earth Biogenome Project. We have seen a pretty significant change in the face of genomes. And so even if you look at some of the marsupial genomes from the very earliest opossum genome that was produced back in 2007, right through the last decade of genome sequencing, we've got better and better at sequencing reference genomes and producing data that is bigger and bigger data sets, which is fantastic because it allows us to ask really key questions. The long read technology like HiFi and PacBio and Oxford Nanopore allows us to actually be able to annotate through highly complex regions of the genome. So our research lab particularly specializes in immune gene diversity, and immune genes are particularly difficult to annotate without long read data sets. And so even in Australia, we've been changing the way that we sequence genome technology. The AusMammal Genomes Project, which was one of Bioplatforms Australia's first initiatives, produced lots of genomes for the marsupial and mammal taxa, and now we're doing a suite of other species. But with these large genomes and this large amount of technology that we're developing, also becomes the age of big data. And this has really been declared as our new conservation superpower. This is an article that was put together by the Australian Geographic at the start of 2021, which really shows how by combining big data sets and cloud computing, we're able to accelerate the types of information that we're starting to be able to investigate using genomic resources. However, saying all that, all these reference genomes are being created and that there's global initiative for it. When we actually looked in 2018 at the number of species that are listed as threatened by the IUCN, which is the global organization which lists the threat status of many species, of the 15,000 species that they've already assessed, less than 0.8% of them had a reference genome back in 2018. We conducted a very similar study last year again, because these genome initiatives globally are starting to accelerate. But even then, with the Earth Biogenome Project and the Darwin Tree of Life, which is occurring in the UK, and even with some of our genome initiatives down here in Australia, the number of individual threatened species which still have reference genome information is still exceedingly small. And this is something that we wanted to work towards changing. So why would you use genomes in conservation? So genomes and genetic data in conservation have always really been traditionally considered as a nice to have. We have very limited funding towards any form of conservation activity. And with that, we need to start making decisions of what you're going to spend your funds on. Until recently, any form of genetic sequencing or even genome sequencing was exceedingly expensive, but that didn't prevent us when we looked at the 200 national vertebrate recovery plans. There's about 80% of those plans have some form of genetic action in them. So there is a recognition by the global, so the Australian conservation community, that genetic information is important to the conservation of species, even if they didn't have a way of being able to facilitate generating that data easily or even being able to interpret it. When we look at the number of those vertebrate recovery plans of those 200 species, there's only about 15% that were using genetic data back in 2019 when we first did this analysis when we were establishing the Threatened Species Initiative. And this is not because managers don't know. Managers know that genetic diversity is important. Conservation managers know that there's a direct inverse relationship between genetic diversity and inbreeding, that having lots of genetic diversity is important to being able to maintain adaptive potential as well as in the fitness of the individuals involved. And really, when we delved into it, the critical issues started because the geneticists and genome biologists sat on one side of the divide. This actually needs to be changed, this diagram, to be a three-way divide. On the other side of the divide was the ecologists and conservation practitioners and policymakers. And somewhere up here in the sky, which would be my new division that I would do, would be actually put the bioinformaticians, because there's even an ever widening gap between the genome biologists and geneticists who produce the data and the bioinformaticians who have the capacity and the capability to be able to analyze it. What we did, however, look at is when we looked at the two sides of this divide between the geneticists and the ecologists and the conservation managers, we've worked out that if we could figure out a way that we could help bridge this gap, genomics are an exceedingly powerful tool. They're not a silver bullet for every conservation issue, but there's lots of different types of issues and matters that we can address by having good genomic and genetic information. And so this is really what the Threatened Species Initiative is designed to do. The Threatened Species Initiative is about bridging the gap, about partnering between geneticists and the genome biologists, along with the bioinformaticians and the conservation practitioners and the policymakers that really need the information to help inform their decision making. So what is TSI? Well, TSI is an initiative between Sydney University, the Federal Department of Environment, Water and Environment in Canberra, DBCA, which is the West Australian government, and Bioplatforms Australia. And it's really about generating genomic resources for Australia's threatened species, but it's not just about reference genomes, it's also about developing associated population data for these different species, but more importantly, we're building an applied conservation genomics portal, which is really an online toolkit for conservation managers to be able to come and have easy access to information pertaining to protocols from sample collection to what types of sequencing can be done and how do you get a hold of the sequencing companies, and then also a bit of a data analysis portal on how you can analyze your population genetics data. And we've been working pretty closely with the Australian Biocommons and Galaxy Australia about how we can better develop tool sets, so people who are interested in generating reference genomes can access a way in which to be able to assemble their genome and annotate it as well. So as I said, the Threatened Species Initiative is part of Bioplatforms Australia, and Bioplatforms Australia is really about generating omics resources, so metabolomics, proteomics and genomics for nationally for Australia. And part of their biodiversity and environmental theme, there's a number of different initiatives that they've run over the years, from the microbiome projects and OMG, the koala reference genome, and the Genomics for Australian plants, and the Threatened Species Initiative is really building upon all the other information that they've developed through this different environmental theme. So the project was first launched back in May 2020, who launches a massive project in the middle of a global pandemic, but anyway, we decided to. So we launched the project. We did an initial pilot project of only 8 species, and those species were very much species that had different levels in the workflow. So some of them had reference genomes, but no population data. Other had population data, no reference genome. Some of them had nothing at all. And it was a range of taxa, from some plants to fish, birds and some marsupials. And we really have steered clear a little bit in this project away from reptiles and amphibians, not because we don't want to work on reptiles and amphibians, but at the same time that we launched the Threatened Species Initiative, Bioplatforms also launched the AUSARG initiative doing amphibians and reptiles. And so we really work closely with the AUSARG team to make sure that we're not doing any duplication of species, but really also complementing one another, particularly in the bioinformatic pipelines. So we got $1 million of support from Bioplatforms and about close to $500,000 from bushfire funding. We did a first call out last year and have supported about 44 projects just through the Bioplatforms funding, and then been able to support more through the bushfire project. We're spending about $15,000 to $20,000 per species at the moment, and we're planning on doing a phase 3 species release, very much based around the new 100 species priority list that's been put out by the federal government. And also the one other thing we've been working on is the development of the online data analysis portal for population genomics. So this is a little mock-up of what the website, which is under development, will look like. We work in collaboration with Genomics Aotearoa, because a lot of the information that we need to develop for TSI here in Australia is also directly relevant to some of the work that they do over in New Zealand. And so we're trying to, once again, rather than duplicate effort, develop something that's cross purposes across the 2 nations. But the idea is that you can come here, there's some protocols around Indigenous engagement, sample collection and extraction, how you manage your data, where you can get your sequencing done, some educational tutorials around what is genetic diversity, why is it important, what should you be looking out for when you're doing translocations and interpreting some of the standardized reporting. And then, of course, the data portal, which is very much around people being able to undertake both population genetics and parentage analysis, understanding some gene flow between populations. But more importantly, the data portal is really going to produce a standardized report. And one of the things we've come to realize is that standardized reporting is really key to helping conservation managers being able to have uptake of this data into the future. And that's because they receive the information the same way. It'll help educate people in the types of information, because at the moment, they get a different format every time they receive a genetic report from academic researchers. So one of the key things in the step when we first started the project is we started sequencing genomes. We have chosen to sequence our genomes using PacBio HiFi and only PacBio HiFi. Early on the steering committee, we had to make a decision rather than diversifying across all the different types of genome sequencing technology, is really kind of lock us down to one, because it's about trying to generate a set of tools that could be easily used by the community to develop resources moving forward. And so we started with PacBio HiFi, worked pretty closely with the Galaxy Australia team and Australian Biocommons. And that's allowed us to develop this workflow that you can now find for HiFi ASM when you go into Galaxy Australia and be able to assemble your genome. Most genome assembly takes less than a day, unless you have a frog. So the frog genome, unfortunately, wasn't going to fit on Galaxy Australia. It doesn't even fit on our machine that we have at Pawsey, we ended up having to put it in AWS and Amazon Web Services in the cloud. And the reason for that is in the end, it was 3 SMRT cells, a highly repetitive sequence of a 6 gigabase genome, which meant that we used 1 terabyte of RAM in order to assemble it. We could probably get away with doing about 512 gig of RAM, but I wouldn't go anything less than that for a frog because they are pretty complicated. So once you've got your genome assembled in Galaxy Australia, the Galaxy team had worked really closely with the Bioplatforms data portal as well. So it makes it much easier now to do data transfers between the data portal when you get the sequence data sent to Bioplatforms data portal from the sequencing companies, and then directly into Galaxy Australia. And that's critical for a lot of organisations who are involved in TSI who don't have on-prem compute that they can pull down the data onto before they can send it up to Galaxy. And that's anyone who doesn't work at a university. So these are the people who work at museums or some of the NGOs or other conservation agencies who are working in this area, but just not really having the compute support. And that's really what we've tried to do, is develop open access support for this community that sit outside of the academic sector. And the reason for that is you'll see shortly when I show the number, the breakdown of where people who are involved in the TSI project come from. So the other thing we've been working pretty closely with the Biocommons on is getting a handle on how to do the genome annotation. We have a Pawsey Nimbus machine that's got 64 CPUs and a license for SGNesh++, which is a software license. And FGNesh actually uses, the default setting is it just uses homology-based approach. You get a gene matrix get it downloaded from Softberry, and then it's usually of a closely related species, and it just looks for homologs. The only problem with this is it's not really that useful for Australian species, because a lot of the species that we have here in Australia are endemic and found nowhere else in the world. And Australia are actually separated from the rest of the global community in geographic time about 53 million years ago. And so sometimes our species here have lots of gene expansions or different types of gene expansions to what is found elsewhere in the world. And the gene matrices that we find in FGNesh sometimes don't really annotate the genomes that well. What we've been doing, however, is also adding our own evidence. So if you have the ability to have transcriptomic data, you have to build your own global transcriptome assembly. And this really uses the predicted proteins to be able to improve your annotation. One thing I will say is if you are going to be using FGNesh on the Pawsey machine, please make sure that you've read the documentation and have built your global transcriptome before you try and do your assembly. The global transcriptome assembly does actually take about a week to 10 days to assemble. And so you end up just wasting time on the annotation machine while you're trying to get your assembly and self set up. The good thing about the genome annotation is it doesn't actually take that long to run. You can run a decent genome annotation in a few days on the Pawsey machine. So once you've got your assembled genome, of course, there's a number of different areas you can release your genome. You can either send it to NCBI, which is one of the global standards, but also what we wanted to do for the Australian community is develop a way that we could publicly release our genomes here that we've assembled that may not be ready to go up to NCBI, but you still want to make them publicly available or available for people that you might be collaborating with or other people that would like to use your genome. So we have become part of the Registry of Open Data on AWS, and AWS is supporting the hosting of those data sets, and they also will pay the egress fee. So it's free to download from the Australasian Genomes site. You can click on the link there and you simply come up with, it's a GitHub page that comes up. You can find your genome, you hit the species data button, and it takes you through to the different genomes. And once you can go in there, you can download the genome itself, the associated BAM files. And for the Koala Genome Project, which I'll talk a little bit later on about, we've got about 206 whole genomes publicly available at the moment on the Koala site. And really, this is possibly the only place that we can store this information, because the Koala Genome Project is producing 450 30x coverage or more genomes, and the BAM files alone are going to be about 700 terabytes of data, which no one really wants to host on their own on-prem system. So that's really a lot of information very quickly about the assembly and annotation pipelines that we've worked together that exist already on both Pawsey and with the Biocommons and Galaxy Australia. So all that documentation is coming together and is getting released at the moment, and that all that's accessible at this point in time. One of the things that we're just finalising is developing the population genomics data portal. And so the reason for the length of time for population genomics to come about is how it works is that you can get a reference genome to align to. So we use a stacks pipeline and you can align your data to any kind of reference genome. And this pipeline, the stacks part of this pipeline will also be available on Galaxy if anyone is interested. But you can pull your genome down from any of the publicly held places, whether it's AWS, NCBI, the Bioplatforms data portal or the like. But the idea is that the end user can go out and collect the samples from the species of interest that they're working on. They can then extract those samples and have them sequenced by a commercial provider. They then get their data back, and then what do they do with it? So the idea is if they can come to the TSI data portal, and that allows them to process the data using a cloud-based portal system. And so we've actually got a project at this part of the project is in development with AWS and Ronan. So AWS, of course, is commercial cloud computing, and Ronan is a cloud architecture company that is a very easy to use interface that you can use with the AWS system. And we have a commitment from AWS to provide us with some free computing for the initial first phases of the data portal. But in the longer term, it's been designed and set up that you can get it from the AWS marketplace. You can basically pay for the compute that you use, and once you shut the machine down, you no longer need to pay to use it. It's actually pretty cheap to run. We can run a catalogue of about 4,000 Tasmanian devil samples and call our SNPs from that and do our population analysis, and it takes about 5 hours and costs us about $30 US in the cloud. So this is the landing page. You can come in, click the Get Started button, and this just gives you a little bit of indication. This is some of the preliminary build at the moment. You can find your Bioplatforms data set. You basically put the link in there and submit it, or if you have an API key that you get from BPA, you can decide whether you want to do a de novo SNP calling alignment, or you can align it to a reference genome. And again, you can either find your Genome Assembly from Australasian Genomes site or NCBI, and then it goes through a series of questions and steps pertaining to how you want to do population genetic analysis. You have to download a template in order to load up your metadata, all those kinds of things. And then at the end, once you've submitted your job, you can actually run a number of jobs at a period of time. You can check out what's happening with your jobs. It'll show you the progress of your data set, how you're doing the method, what reference genome you're doing, any downstream analysis. And one of the other factors we've asked them to put in here is what is your running cost of your job progress at this point in time, which we think is pretty important. You can also come in here and realize that you might not want to keep going with that job. You can simply cancel it and kill out the machine. So this will become quite versatile. And the aim is to have this data portal up and running for beta testing, hopefully by May this year. And after we've beta tested it a bit, we'll then be rolling it out for the TSI community. So coming back to this, once you've done your you've gone into the data portal and you've got your standardized report, the end user can use that report and interpret it with some assistance initially, and hopefully over time will be able to start to interpret the information better themselves. And then you can actually use that information for your decision making. So you can use it for translocations or captive breeding or whatever the types of questions that you've asked of it. We know this system does work. This is actually me collecting Tasmanian devil samples out in January in Tasmania, and we can run through this whole process and move animals for translocations in June. So we've got it down to about 4 months when it's working well. And that a lot of the work that we've built into TSI is built on the back of the work that we've been doing for quite a long time with the Tasmanian devils, and to a lesser extent, some of the other species such as bilbies and orange-bellied parrots here in Australia. So for the conservation managers, there is a vast array of questions that you can ask and answer using genomic information. And I'm not going to go through each of these now, but it is ranging from just population diversity and inbreeding and how populations are different or adapting to climate change, looking at relationships if you're running breeding programs or translocations. And it also works, the portal works for both DART-seq data and DDRAD data that we get from AGRF. It works on large open populations, small, highly inbred populations, and we're working with the plant guys to try and make sure that it works for some of the plant species as well. So really, what does it mean once you have a genome? So once you've got a genome, you can investigate questions that are very much pertaining around neutral diversity. And we talk about neutral diversity as being the non-coding regions in the genes that are under a faster rate of mutation, which really allows us to look at differences between populations, population diversity and structure, and allows us to also be able to determine parentage analysis, excuse me, which is very important when you're doing captive breeding programs. So the other things that you can do once you have a reference genome is actually to do more functional based questions. So in our research group, for us, it's very important to have an understanding around immune gene diversity and disease resistance and recovery. We work with quite a number of species that have disease, but you can also use it to characterize gene families such as reproductive genes if there's an issue around fecundity. And we're starting to investigate some questions around heat tolerance and heat shock proteins and the ever-changing climate that is coming. And we know from some of the pretty strict climate modelling that we have here in Australia, particularly in New South Wales and Northern Queensland, of how much the landscape is going to change in those environments as the climate continues to change. The other thing that we can also use genomes for, HiFi genomes are particularly good at this. You can actually get a mitochondrial genome just from your HiFi data set. There's a pretty good pipeline that allows you to work out the mitogenome. And from the mitochondrial genome, you can actually now develop species-specific eDNA markers. And this is something that we're just testing out in our lab at the moment with some of the endangered species that we work on, to see whether or not, rather than just saying whether it's an amphibian or a reptile, we can actually start to nail down the exact species using these marker sets. And also you can develop species-specific target markers, either using mass arrays for scat samples, or we can do targeted gene probes using MyBaits to be able to pull out really specific genes. And we can do hundreds and hundreds of samples if you're just looking at specific genes. And none of that, other than neutral diversity, is possible without a fully annotated reference genome. So when we look at some of the species that we're doing in the Threatened Species Initiative, these are some of just the little critters. You can see we do a wide range from freshwater crayfish through amphibians, some of the mice, parrots, plants, fish, marsupials, reptiles. I think I've covered everything there. And as you can see, most of them are either endangered or critically endangered. And these are individual populations which are actually going down. And what the aim of the game is to see whether or not we can provide enough genetic information which can contribute to some of this species recovery. So at the moment, we have 62 species in 40 projects. You can see on the left-hand diagram here, this is the threat status of the species. We have a mix of critically endangered, endangered and vulnerable species. DD, which is the grey one, is data deficient. So these are species that we just don't have enough information on to be able to even assess what their threat status actually is. And the black species you can see here is we have a couple of extinct in the wild species. So these are species that no longer exist in the wild at all. They're only held in captivity or in human care. And so really, these are the species that if we can't figure out how to put them back into the wild, they will go down into extinction, and then we will have lost them forever. On the right side, you can see the diagram by the taxon group. We've got a pretty even breakdown in the different taxon groups. You will acknowledge that there's a very large number of plant species. That's based on a couple of projects that are doing a wide variety of plants. But it's also kind of representative of what we know about threatened species here in Australia. There's approximately 1,400 threatened plant species in Australia, as opposed to only about 500 animals, vertebrate animals. One of the things that we haven't done a lot of work on to date, which is something that we'd be very interested in working on more in the next phase of TSI, is starting to look a little bit more around the invertebrates and how to better provide genetic resources for invertebrate species. So it is a national consortium. It's got quite a range of spread of people. These are the number of species in each state. The large number of species that are getting done in New South Wales and in Queensland is an artefact of the bushfire funding, as of course that was targeted to where the 2019-2020 bushfires were. But we try to get species that were representative across all states of Australia, working pretty closely with the different First Nations and Aboriginal people to make sure that we're taking into account what is important to them, as well as helping local communities answer some of their key questions. But you might remember when I was talking a little bit about why it's so important for all of our compute resources to be outside of the academic sector. You can see here only about a third of the people actually involved in the collaboration of TSI come from academia, where the rest of them either come from the yellow is NGOs, conservation organisations such as World Wildlife Fund and the Australian Wildlife Conservancy, and the other almost half of our collaborators are coming from government agencies, and these are people who have an interest in using genomic resources to inform their policy and their decision making. They're really keen to use the latest technologies, but they don't actually have compute access through their current systems unless they have an affiliation with an academic organisation. And so that was one of the key areas that we really wanted to ensure with Biocommons and Galaxy Australia and Pawsey, that we can provide this access to them. So these are some of the species genomes that we've actually been using with Galaxy Australia and starting to annotate on the Pawsey machine. You can see we've done birds. Birds are really easy, got short little genomes. They're only about 1.3 gigabases. And we've done a reptile. The frogs are the interesting one. So this is most of these species that you can see on the screen here are all listed as critically endangered here in Australia, except for the ghost bat. That's only listed as vulnerable. So of all these critically endangered species, the Kroombit Tinker Frog, which is this lovely little purple guy here, has been the biggest problem in the fact that we weren't able to get an adult frog. They have no idea how many is left in the wild, but there's 3 in captivity up at Currumbin Wildlife Sanctuary. They did have a number of tadpoles. We were able to euthanize a couple of tadpoles. We had to skin the 2-centimetre tadpole in order to be able to get enough DNA from it to do the HiFi sequencing. That frog ended up requiring 3 HiFi smart cells, and that little 2-centimetre frog is the frog that we had to do in AWS with a terabyte of RAM in order to be able to assemble the genome. And we've just completed the annotation on the Pawsey machine just last month. So we're now starting to characterise some of the antimicrobial peptides in that little frog. So once you've got all these reference genomes that we've generated, what really is a genome good for? And I've given you some indication, but I thought I would just take a little bit of time this afternoon before we finish up and showing you a few case studies about what we can actually do when we've got a genome. So this, of course, is the Tasmanian devil. The Tasmanian devil story is very well known. In the last 20 years, we've lost about 80% of Tasmanian devils across the entire state of Tasmania as a result of an infectious clonal cancer, devil facial tumour disease. Devils, in fact, have 2 forms of infectious clonal cancer. And as you can see, the disease is really swept from the northeast, where it was first discovered back in 1996, and swept westward and southwards across the state until most of the state now is infected. So what have we been able to do for devils with reference genomes? So the reference genome for devils was first published in 2012 by Liz Murchison. And we were able to directly use that information and feed it into our conservation management. So we had the devil genome was produced, as well as transcriptomes, both from the devil itself and from the facial tumour disease. That allowed us to annotate a range of immune genes, which are highly variable regions and repetitive regions in the genome. And a recent study that we've just shown is that automated annotation pipelines are only pulling out about 30% of all the immune genes when they're doing the automated annotation. And actually you need to do manual annotations of the immune gene region if you really want to be able to characterize this region well and use that information for any kind of purposes moving forward. So we've used that to really investigate the relationship between the cancer itself, the devil's immune system. We've looked at these are antimicrobial peptides, which have very potent antimicrobial activity. These peptides are produced in the pouch and the milk, the mother's milk in the marsupials, and we know they've got very, very strong properties, and they actually kill DFTD. We've also been conducting studies into different types of microbiome, and we used the devil genome to be able to develop a blocking primer, so we can only pull out the 16S RNA from the microbiome, and the virome we've done now as well. We've done some genome-wide association studies using the reference genome and some resequenced genomes. We've also then used a whole suite of this information to develop species-specific population genetic markers, which allowed us to undertake the translocations. So essentially all this information that we produced in a very short period of time, when I said it, took about 3 minutes. That's probably 10 years worth of work done there, is feeding into those management decisions. And based on what we were able to do with devils, we can do a very similar thing again with orange-bellied parrot. Orange-bellied parrots are one of Australia's most critically endangered parrots. We got down to 3 females and only about 10 males in the wild a few years ago. Been doing a large amount of translocations for this species, but this species suffers significantly from citizen beak and feather disease virus, and that affects the birds' feathers and their plumage. And this species migrates from southern Tasmania to Victoria every year and back again. And without having decent flight feathers, you're not going to survive the migration journey. And so what we've done with those birds is we actually generated some DART-seq data, and we've been using that information to really start to understand how we should be translocating these birds. We also developed some primer sets just for the TLR, which is one immune gene family. This is all prior to us developing the genome. So we've actually just finished the assembly and annotation of orange-bellied parrot genome. We now have the full assembled annotated genome, and working with a suite of resequenced genomes to really get a handle on what's happening with diversity at a genomic level for this species. Because we know that there are no other populations located anywhere in the world that we can obtain new diversity from. And if the species is lacks too much diversity at the immune gene region, then the likelihood that there will be long-term persistence is really minimal, because they don't have any variation in order to adapt to the changing disease environment. And that then lends to conversations that we're starting to have around, well, how much genetic manipulation do you need to be able to do to be able to save a species and inform the management decisions long term? So watch that space for what's going to happen with OBPs. One of the other things that we do in our research group is we do a lot of work around antimicrobial discovery. We are part of a very large centre of excellence that started last year about innovations in peptide and protein science. And by using the reference genomes of the suite of different Australian vertebrate species that we work on, we can use these genomes to characterize these antimicrobial gene families. So for example, marsupials have quite a large number of what we call cathelicidin genes, as opposed to humans where we only have 1 or 2. And we see a lot of antimicrobial activity in the skin of frogs as well. And so types of things that we can do is we can develop these antimicrobial look at these peptides that we know kill DFTD and devils. The koala peptides kill koala chlamydia, which is a disease severely impacting koalas. And then, of course, we want to get a better understanding of how these peptides could potentially be used to combat chytrid fungus in frogs. And so really there's been no new forms of antibiotics for humans even in the last 50 years, and antimicrobial resistance is really becoming quite prominent an issue for people as well. And now what we want to understand is whether or not these antimicrobial peptides that we find from nature, can they actually be used as a new way of helping assist antibiotic resistance even in humans. And so you can mine both the genome and the transcriptomic data for these short peptides, and then use that information to help develop ways in which you can test the toxicity and determine whether or not you should be using these peptides. So the last case study, of course, is everyone's favorite furry animal, the koalas. This is the Koala Genome Survey, which we started just last year, an ambitious project to sequence whole genomes of over 450 koalas across the species range, all the way from northern Queensland right through Victoria and into South Australia. And where it became so particularly important is if we look at New South Wales, the green areas that you can see on the map are the areas in New South Wales which they call areas of regional koala significance, so areas where we know that there's always been traditionally large populations of koalas. And the black grey areas you can see on this, these are all the areas that burnt in the 2019-2020 bushfires. And most of the genetic data for koalas has occurred up here on the north coast, or around about Port Macquarie and a little bit around Sydney. But there were large areas of the state which burnt, where we know koalas have existed, but we just don't have any genetic data for them. And so we started the large koala genome survey. It's supported in funding from both the New South Wales government and the Australian federal government, as well as support from Ramashotty and AWS and Illumina in regards to the sequencing and cloud support for hosting these genomes. We've received over 600 samples now. Not all biological samples that are collected are suitable for whole genome sequencing, particularly when you're going to a depth of 30x. We're getting most of our genomes are getting up to about 45x coverage. So we need pretty high quality DNA from the samples, but to date we've sequenced about 260 samples already, and they range from up here in Atherton in Queensland all the way through Queensland and Victoria, and we just received our South Australian samples the other day. The most exciting thing about this project for me is the massive uptake that we received from the koala community when we first asked them about joining this program, and the fact that we make the data publicly available as soon as we've sequenced it. So we're aligning the data through the Illumina Dragon pipeline. This is a fantastic pipeline that they released last year. It's based on GATK and does the alignments of a 30x coverage genome against the koala reference genome, which is a high quality long read HiFi genome. And it does it in 72 minutes per sample. It calls the VCFs against and aligns the BAM files. So it makes it pretty easy for us. And it is quite seamless because the data is sent directly from Ramashotty up into the base space with Illumina, where we can do the alignments and then we can pull down the genomes and the data that we can. So again, it's very similar management questions to the ones I've already talked about for the suite of other species. The questions that most of the people around Australia have is like, you know, what's happened as a result of the megafires and what's the risk of genetic extinction now for a number of koala populations. Koalas have just been listed as an endangered species in the ACT and New South Wales and Queensland. I'm really starting to get a handle on what is the genetic basis of resilience to disease for these populations, and potentially drought as well, and what's the links between functional genes and population dynamics across taste receptors and food type, heat tolerance and phenotype, immune genes and disease susceptibility, and even the reproductive genes and fertility, and even considerations for translocations as well as managing captive breeding. So really, I just wanted to finish up today by saying that the age of genomics is well and truly here. It's well and truly here for conservation and biodiversity, and it's going to be something that is essential for us in combating the loss of biodiversity. As I said, biodiversity is underpinned by these three pillars, and the Threatened Species Initiative is very much around developing genomic resources for these threatened species that we have in Australia, using the Conservation Genomics Hub that we're trying to develop to make it resources more accessible, easily accessible, working in collaboration with the Australian Biocommons and the rest of their community to find support and compute access for people who are working in this space. And really using this information, it has a wide range of applications for management, policy, and even understanding the ecology of species. I once asked one of my partners in the Devil program, you know, what has having had all this genetic data for devils over the last 10 years done for them in the management space? And they said genetic data, having genetic data available for management of a threatened species is really the difference between flying blind or flying with a navigation system. This is not just me at all, or Sydney University. The Threatened Species Initiative is a very large collaboration with many, many partners and parties, of which none of what we are achieving today would be remotely possible without all these people contributing to the project. And so really, we should acknowledge all these communities. And so what I wanted to finish up with today is this image, and I love this image. This is a photograph that was taken in 1968, and it was the first photo we ever saw of our Earth. And it really, to me, depicts how fragile the planet could be and what human ingenuity can do when we really apply ourselves. And in the 50 years since this photo was taken, we have genome technology, we've got artificial intelligence, we have machine learning, we have cloud compute, the internet, mobile phones in your hands. And we've also had 350 species go extinct in the same 50-year period. And so what we now need to do is focus on how we can always take this human ingenuity that we've developed and do something better for our planet, because I would really like to think that in 50 years' time, it is a different world to the one that we're now facing. So thank you very much for having me today, and I will pass you back over to Christina.
Thank you. What a way to end. Thanks so much, Carolyn, for the excellent overview you've given of a very complex network of projects that are doing such important work in related to threatened species. So now we do have time for some questions. Please type your questions into the Q&A box in your dashboard, and we'll do our best to get to them. We've got a great question to start with though, Carolyn, and one that asks you to think ahead, not into the next 50 years, but into the next 10 years, perhaps. In your opinion, what will the connection between genomics and conservation be like in 10 years' time?
Oh, if I had my way, everyone will be making decisions for anything around translocations or revegetating habitat, captive breeding, and planning where we're going to find climate refugia for species will all be underpinned by genomics, because I've made a career of trying to reverse decisions that were made by moving animals or planting trees or revegetating habitat without that kind of information at hand. And now we have such great sequencing technology. We really have an understanding of how making ill-informed decisions can have long-term consequences to conservation. Classic example is planting a whole bunch of revegetation. I think it was like something like 20,000 trees that no one had checked whether or not they were clones, and they were all clones of one another. So we basically planted a forest of trees that were going to be sterile when they reached maturity in 40 years' time. So it's simple things like that. You know, we now have the information to be able to inform what we do. And I think that's really where I'd like to see it, where it's not a nice to have or an add-on or something that's really difficult and complex, that it just becomes a part of the everyday management process, the same way we use camera traps and satellite tracking and starting to use drones. We should just start using the technology.
Hear, hear. Another question for you. How do you choose which sequence method you will select? Is it a matter of weighing up cost and read length for different options? How do you make that decision?
So this is someone who's asking me how to choose between HiFi and nanopore, because that's pretty much where the questions usually start. I wouldn't go short read data ever again. It's too hard to annotate complex immune gene families. That's the one big lesson we've learned. We had to make a decision at the start of TSI, and one of the reasons we chose HiFi was simply there was some well-established bioinformatic pipelines. So it does cost a little bit more, but I think there's been some investigations by the AusArg team as comparisons between HiFi and nanopore, and HiFi really meets the needs that we require for what we're trying to do in the conservation space. It is a difficult decision to make, but at the end of the day, it comes to cost and efficiency and ease. And cost isn't just how much it costs to do the sequencing. The cost that people tend to forget about is how much it costs to do the compute at the end, and the storage of the data files that you produce. And that was part of the reason why we went with HiFi, is because at the end, when you calculate all that, it became a little bit more efficient for us. In relation to the population genomic space, that's always a difficult one. We use DartSeq or ddRAD. Those are the two common methods used here in Australia, because it's reduced representation sequencing. It doesn't matter which one you use. The results you get from both are roughly the same. The only thing you can't do is use them interchangeably. So you can't sequence half your samples using one method and then try use another method, because the enzymes, cut enzymes they use are different. So the only really big caution to people is choose your method wisely and stick to it, because you're going to be stuck with it for a while.
Okay, thank you. I think we'll just take one more. We're running out of time. We've got a question here about, is it challenging to build a genome for a species that largely relies on historical samples only?
Yes, it does. So historical samples, because they traditionally have quite degraded DNA, makes any form of long read technology quite complex and quite difficult because the DNA is simply too fragmented. I know that there are teams globally trying to work on how to resolve that issue, but at this point in time, yeah, without the long kind of DNA strands, insert strands for the sequencing, it does make it very complex to use long read analysis for historical samples. But saying that though, you can use short read data and then try align it to a long read genome of a more closely related species. I have heard of some folks doing that, but I'm not quite sure what it looks like at the end.
I might just squeeze in another topic there. Live questions come into. You briefly hinted toward the prospect of manipulating genomes to create artificial genetic variation in this work. Is it possible, and has it been done?
Well, it's possible to do in a chicken. So that's a good start. I personally, and Kathy Belov, who I work with, we would, if we can't find any genetic variation in an orange-bellied parrot, I would like to try and find some diversity in historical specimens and see whether or not we can gene edit it in. But the problem with gene editing technology is you need artificial insemination and assisted reproductive technology to be working for the species at hand. And that kind of ART assisted technology is different for every single species. And so that's where it becomes very complicated very quickly, because it works in a chicken does not necessarily mean it's going to work in a parrot. So I don't know if I'm going to go down that road yet, but I think it's a very interesting concept.
Indeed. Well, that is all we have time for today. So once again, I'd like to express our thanks to you, Carolyn, for taking the time to talk to us today.
Great. Thank you so much for having me.
So this webinar was part of a series of bioinformatics training events organised by Australian Biocommons. You can find out more about upcoming events like webinars, workshops and other events too on our website, and you can watch recordings of previous events on our YouTube channel. If you do go to our website, you'll see that our next webinar is about the protection of genomic data and the Australian Privacy Act. So that talk will ask the question, when is genomic data personal information? It's going to be delivered by Professor Mark Taylor from the University of Melbourne on the 6th of April. So we'd love you to register for that. As you leave the session today, please take a moment to fill in the quick little survey that pops up on your screen, and this will help us with our planning for future events. Finally, we'd like to acknowledge that the Australian Biocommons is enabled by NCRIS via Bioplatforms Australia funding. Thanks so much for watching today, and we hope that you really enjoyed the presentation and that you'll join us again in the future. Until next time, goodbye.